Okay, hi everyone. Now today's video is based on our blood transfusion and blood grouping. Okay, so in this we will all talk about our transfusion of blood and how the blood grouping concept came into existence. Alright, so you all have seen that it becomes necessary sometimes to inject blood into the body of the patients who are undergoing surgical operations. Okay, so when we do this, that is nothing but it's called blood transfusion. Alright, so what is happening actually in this blood transfusion? So a blood is going to be taken, okay, from the healthy person. So the person who is going to give us the blood, okay, the one who gives the blood is called our donor, and that blood is introduced, okay, through one of the patient's veins to the particular person. Okay, so the one who receives this particular blood, okay, they are known as your recipient. Alright, so we now know what are the donor, which one is known as a donor and which one is known as your recipient. So whenever we carry out blood transfusion, there are two important things that we have to remember, that one will be the donor and the other one will be the recipient. So you all know that now there is a donor and a recipient and how the blood transfusion takes place. But do you think we can take blood from any person and then transfuse it into anyone else? Is it so possible? No, that is not possible. Blood transfusion is very specific. That means there has to be some kind of compatibility. You cannot just take blood from anyone. Okay, that is why you all have heard that there are different types of blood group among yourselves only. If you discuss, if you are 5 to 10 people around, if you ask them, there will be different of your blood groups, okay, and you will have A, someone will have B, someone has O, someone has AB. So there are different blood groups and you cannot just take blood from anyone. Okay, there has to be certain compatibility for taking the blood. That means blood transfusion is highly specific and it has to be compatible between the donor and the recipient. Okay, so if we talk about in this blood transfusion, okay, we will move on first with that what is your blood transfusion. So we all know that now blood transfusion is nothing but it is actually the transfer or transfusion of blood from a healthy person, that is a donor, which is introduced through one of the patient's veins. All right, and this is what it is a highly specific, okay, process. All right, and transfusion will take place between the compatible people, that is the host and as well as the donor. That means the donor and the recipient has to be compatible. Okay, so now we move on to the next slide. We can see out here how blood transfusion is done, and you have been to the hospital and you have always seen this view. So this is all about a blood transfusion. Okay, now we cannot transfuse blood just like that with anyone. Yes or no? We just say that we cannot give blood to anyone. It has to be compatible. So why there was this need? Okay, so it was the concept of blood grouping. So this discovery of your blood grouping system, okay, who did it? So it was Carl Landsteiner. Okay, he was the one, right, who brought the concept of blood grouping into existence. But how? What did he do? Okay, so he was carrying out the studies, okay, on the RBCs of human beings, and then he found out that there are certain specific proteins on their surface. Okay, he found out. So while studying what? While studying the RBCs of the human being, he found out that there were certain specific proteins that was present on the surface of the RBCs. Okay, so he started discovering them. So then when he took blood samples from different individuals, he found that some of them, they had, okay, you have to remember some proteins, okay, which was somewhat like this. You can see some of their RBCs from individuals, they showed the presence of this type of surface proteins, and some showed this also as well as this also. So they had both the types of your surface proteins on the RBC. But there were even certain cases where he found out no such surface proteins on the surface of RBC. So when we look at it now, it is very clear that the individuals who do not show the presence of surface proteins on their surface of RBC, actually they are blood type O. So he named them as blood type O. Whereas there were certain individuals who showed the presence of your surface antigens, that was your proteins. Those surface proteins were given a name, and that was the antigen. So they had surface antigens of this type, this particular type. You can see it's yellowish in color. Okay, just to give you a brief idea. It may not be yellow, it might be looking something different. Okay, but just to show you and help you imagine things, they have showed you in this color. So therefore they named them as blood type A. Okay, whereas there were certain individuals whose blood sample, when it was collected and the study was carried out on RBCs, they show the presence of surface protein which was of this type. So that was the surface antigens were of this particular structure, whereas there were certain individuals which showed both of them. They had this surface protein also as well as that surface protein also, or we can say this surface antigen also and that surface antigen also, both A and B. So therefore he named them as AB. This one was named as A and this one is named as B. Alright, am I clear? So based upon the surface antigen or the surface protein, the RBC shows the blood grouping system came into existence, whereas the one which do not show any surface protein on the surface, they are called your blood type O. Is it clear now? I hope it's clear now. So this is what it is all about, your blood type. So therefore the concept of this blood grouping system came into existence by the scientist, very well known scientist, that is our Karl Landsteiner. Okay, and it was he who developed this blood grouping system, where he was carrying out study on what? He was carrying out his study on the surface of your RBCs, what type of proteins, what are the structures present. So when he was doing and he's carrying out his study and experiments, he finally found out that there were certain structures that were present on the surface of RBCs of many individuals, while some of them do not have any such kind of surface structures. Okay, so therefore he named them as surface antigens based upon the particular structure. So let's say in some of them there was no, so he named them as blood type O. Whereas in some of the individuals they showed this type of structures, so it was blood type A. Some of them showed this rounded structures on the surface, so it was blood type B. And some of them showed both the triangle as well as this rounded structure, so it was a mixture of A and B. So that's why it's a blood type AB. That means this one had both the surface antigens A and B present. This has the surface antigen only B present, and this has only surface antigen A present, and out here no surface antigens are present. Okay. Alright, so we can see out here, so red blood cell type, okay, we have our group A, we have our group B, we have a group AB and group O. So this is a blood grouping system, group A, group B, group AB and group O. So you can see, okay, in detail out here. So this is the actual structure, so red blood cell type, look out here. Look at the structure present out here, outside. So he named these structures, okay, as surface antigen A. This one was surface antigen B, and this one was mixture of both it had. So surface antigen, both A and B are present out here. Whereas in O, you can see no such surface antigens are present. Okay, so if after that, while carrying out his study on the antibodies, okay, he even studied the plasma. When you study RBC, obviously when you take blood from the person, you have the plasma as well as the cellular element. So while studying the plasma, he was able to find out that this blood group A, the individuals with blood group A, okay, so their surface antigens were A. Yes or no? They had antigen A on the surface of the RBC. So the antibodies that were found in the plasma was antibody B. Just remember, it will be the opposite. It was found that the RBCs having the surface antigen A, their plasma had the antibody B. Whereas the group B, that means the blood group B, those RBCs which had the surface antigen B, their plasma, okay, they had the antibodies A. Alright, and whereas in group A and B, you can see that both are present. So the plasma had no antibodies. What were there? No antibodies were present in case of them. Whereas in case of a blood group O, antibody A and antibody B, both were present. So once again, if we see, you look at the structures out here, these all are your surface antigens. Whereas in O, there is no such surface antigen present. Okay, so you can see According to the structures, he named it as antigen A is present out here, surface antigen B is present out here, and out here surface antigen A as well as surface antigen B is present. But the plasma of this A blood group, they showed antibody B. The plasma of this blood group B showed antibody A. Whereas in AB, since both of these are present, so there was no antibodies in the plasma. It's not possible. Whereas in O, since none of them are present, so therefore there was both antibody A and antibody B in the plasma. Okay, this table is very, very important. And if you talk about the surface antigens that are present, you can see A antigen, B antigen, A and B antigens both out here, and out here none are present. That's it. That is it all about your ABO blood grouping system. So there are two blood grouping systems. Please remember that. One is your ABO blood grouping system and the other one is our RH system. Okay, this is another way of having a blood grouping. So we just now discussed in the previous slide that this is a blood group A, B, AB and O based upon the surface antigens that are present and the plasma containing the antibodies or not. So you can just remember one thing, in case of your this blood group AB, both your antigens A and B are present. Okay, whereas in case of a blood group O, none antigens are present. But if you talk about the antibodies in the plasma, in case of your AB, none are there, whereas in case of a blood group O, both antibody A and antibody B is present. Antigen antibody concept is very, very important. Okay, so antigen is this protein structures that are located on the RBCs, anything that is protein, and antibodies are certain release of your certain structures into order to fight. Okay, they will act as your fighting material. So you know that the WBCs will produce your antibodies, so it's according to that. Just remember this much for now. And the second type of blood grouping system is our RH system, where we will see that RH blood group means what. Now you all know, whenever I ask you what is a blood group, so you say A positive, B positive, AB positive, A negative, B negative, AB negative, or you say O positive or O negative. Don't you say that? Yes, of course you say that. So this positive and negative fact which is there, it comes from the RH blood group system. Why we say positive and negative? So apart from the surface antigen that is present on the surface of your RBCs, there were certain other structures or factors that was found in the individual's RBC surface. So those were known as your RH factor. So the individuals having this RH factor present on the surface of the RBCs, they were termed as a positive, so they were RH positive, whereas the individuals with no such factor on the RBC surface were named as your RH negative. So apart from this surface antigens that are present, there was the presence of structures like this also. Okay, along with what? Along with this surface antigens, the blood groups of many individuals showed the presence of structures like this also. So those blood groups along with the surface antigens, if they show the presence of RH factor, these structures, then those blood groups are all positive. That means AB positive, A positive, B positive or O positive. But if the blood groups they do have this surface antigens, they have the surface antigens, they are A, B, A, B, O, they have the surface antigens. In this case, out here there is no antigens. So if they have this structure, apart from all this, if the RH, this factor is not present in them, then they are termed as your RH negative. That means that blood group becomes negative. A negative, B negative, AB negative, O negative. You all have heard about. So that positive and negative comes from this RH system. So this RH system, the blood group of most people contains actually the substance called your RH factor. RH actually is coming from where? It actually means it is coming from your rhesus or common monkey. In which this factor was first discovered in a monkey. So that's why we are giving an RH system over there. So when the blood of an individual, shows the presence of this RH factor, that means that blood group has a positive charge, and if they do not show that, they are termed as a negative. So therefore you have the positive and negative blood grouping comes from your RH system. So when the blood of such an individual, let's say the RH positive, is transfused into a person which lacks this RH factor, that is RH negative, then the blood of the recipient will develop an antibody Okay, means it will act, that particular blood will act as a foreign substance. So therefore our body, if it is being transfused in a person, let's say, so they will develop antibody, and then what will they do? They will just act against it. Okay, and then within about 2 weeks of transfusion, the person will get sensitized. Alright, so we do not have to go in so much of detail. Just remember that RH system, there's, if the blood group shows the presence of RH factor, it's your RH positive. If it does not show the presence of RH factor, it's RH negative. And the most important thing is your ABO blood grouping system. Which surface antigens are present, and what is the antibodies in the plasma. This table is very, very important. These are the blood types. Why they're classified? Because of on the basis of their presence of the surface antigens. And who discovered or gave the concept of blood grouping system? It was Carl Landsteiner. That much we have to remember. Okay, but when you go for higher classes and higher studies, you will be studying about, okay, the RH factor in more detail, but for now it's not required in so much detail. So I hope you all understood the concept of your blood transfusion and blood grouping, where we talked about what is blood transfusion, you know that, and then we even talked about our blood grouping system. That concept of blood grouping was given by who? These were the blood types. Okay, and then we discussed this table. Now I would like to give you another important knowledge out here, that is, you have to remember, since this blood group O, okay, it does not have antigens on their surface, neither A nor B, nothing, so they are termed O. Okay, so they are found to be universal donor. That means they can donate blood. All right, what is that? They can donate blood to AB also, to B also, and to A also. But this blood group O cannot receive blood from these three. It requires O. That means the individual with O blood group only will be able to give them the blood. Okay, and that positive negative is also there. We will talk into later. But remember that this O positive or O blood group O, those who have, okay, they can donate blood to the all. So therefore they are universal donor. Okay, whereas your AB, it can receive from all, because it can donate to also. O will give it to this, and see, this B will also give, A will also give. Why? Because it has both the surface antigen. So if the blood group, a person with the blood group B gives it blood to AB, then they can take, and out here A also. So therefore this is a universal recipient. So it can receive blood from any of these one, but it cannot donate to all. Whereas this O can donate blood to all, but it cannot receive from the all. That's it. So universal donor, which blood group it is, and which blood group is your universal recipient. That is, we have to remember it properly. Okay, so I would like you all to just take a brief of this table if you have time, and you can just jot it down. I'll give you just 2 minutes for this, and then I'll show you the diagram for your RH system also. So I think you have understood whatever I taught you in today's class, and we hope to meet again with another video, right? So if you all have any doubts, you all please do let me know, okay, in the class where we can discuss. So this was all about your blood transfusion system and your blood grouping. Thank you, class.